thrown away, which you're going to come back on the 4th to make up for it. So I'll talk about those after. However, the unknown is going to be on week 9, and I'm not going to be here on week 9. So you'll take your lecture final, and Dr. Cindy is going to proctor it. Then when you come to the lab, Dr. Rafiq is going to be here. You're just going to practice for the practicum. So I'll do the explanation. So because before the practice, I usually explain. I've already posted the practicum details on Canvas. So I'm going to explain what to expect so you have a very clear idea. So when you come back on the 9th at 8:15 on Tuesday, you just start practicing. And if you have any questions, Dr. Rafiq will answer. Okay? And then on that Thursday, Dr. Cindy will do the practicum. Okay? So let me go over the practicum. So this is the page that I gave a handout I gave everyone. So all of these techniques have been posted also. Again, the practicum is only on the techniques, not the results. So we're just going to focus on the techniques, and mainly the purpose of the techniques. Those are the questions that will be asked. So you're going to have, first of all, everyone is going to stay outside, and people are going to come one by one. When you come in and you wash your hands, you put your gloves, you're going to have a beaker. We're going to have a beaker up there like this, and you're going to just take one of the techniques out. You're going to write your name and the title of the technique, and then you're going to put it back in there, so the next person will have the same chance of getting the same technique. So everyone will have, the same chance. So, and then once you perform, you have 5 to 7 minutes to perform, a couple of questions, and then you're free to go. Okay, and then the next person comes in like that. So let me go over what to expect for each of the techniques, and then types of questions that you'll be asked. So I'm going to skip gram staining, that's going to be with the other stains. So I'm going to start with the aseptic technique. What's the purpose of aseptic technique? So you want to make sure that you're not contaminating, because once you open these, medias are like magnets. They're going to attract all the organisms in the air, surrounding anything, any of the instruments, so you're going to have to flame everything. You're going to kill everything because you don't want to misdiagnose, right? Because if that's your clinical sample, you just want to just grow your pathogen, nothing else. So if you get the aseptic technique, we might ask you to inoculate a slant using aseptic technique, inoculate a plate, pour a plate. Any of those techniques you can be asked to do the aseptic technique. Okay, so that's for the aseptic technique, and the question would be what I just asked. If you have Kirby Bauer, so what's the purpose of the Kirby Bauer? For the diffusion susceptibility. That was it was the antibiotic discs. So what was the purpose? To figure out which antibiotic is going to work on your pathogen or on your bacteria, right? So what media did you use? The Mueller-Hinton. So the Mueller-Hinton is going to be out here plus the nutrients agar. So you need to, if you get that one, you're going to utilize Mueller-Hinton. Otherwise, you will lose points. Okay. And the Mueller-Hinton has ingredients that promotes the growth, so that the results would be clear. How do you measure the results, the susceptibility? Why?
The circumference of the clear space.
The diameter or the circumference?
Oh, the diameter.
The diameter of the zone of inhibition, which is the clear area around the disc, right? And do you measure it? How do you measure that? In millimeters, right? Okay, so that's basically for the Kirby Bauer. Now, as far as the strict plating, we have 3 different strict plating. We have continuous, quadrant, and radial. And I have all 3 of them on the board, and I have posted the demo for that too. Which one is for pure cultures?
Continuous.
Continuous is pure. Those 2 are for isolation, right? Isolation. And what do you want to isolate if it's a clinical sample, if it's a urine sample from the patient? You want to basically find out what the pathogen is, right? So you want to separate the pathogen from, or isolate the pathogen from which organisms? What else is in the urine?
Natural
the normal flora, right? So you want to separate or isolate that pathogen from the normal flora. So there are two isolation techniques that utilizes the surface area, quadrant and radiant. In quadrant, you're going to flame and then let it cool and then take the sample and you inoculate the first quadrant. Then you're going to kill, because remember, this is to dilute, to isolate. So you need to basically make it less and less. So do you take more? No.
No.
So that's one of the biggest mistakes. People keep taking more. So always think about the purpose. So you're going to put your sample aside, you're going to flame, let it cool down, and then drag from the first quadrant 1 time, twice, 3 times, 4 times, and now come back. Then flame, let it cool down, and then drag from the second to the third 1 time, twice, 3 times, and now come back, and then flame, let it cool down, and then drag twice from the third to the fourth. So hopefully in the fourth quadrant you have those individual colonies, and one of them is the pathogen. Okay, versus in the radiant, which you didn't do, it's more time consuming. And the way it is you flame, let it cool down, and take your first sample, which is the same as in quadrant. You inoculate your sample. Then you're going to put away your sample, you're going to take your loop and you're going to flame, let it cool down, and you do 1 streak. Again, flame, let it cool down, do a second streak. Flame, let it cool down, do a third streak. Flame, let it cool down, do a fourth from the first quadrant to the second. Then flame, let it cool down, and from the second to the third, you're gonna streak. Flame, let it cool down, do a second streak. Flame, let it cool down, do a third streak. So 4 streaks from the first to the second, 3 streaks from the second to the third. Then again, flame, let it cool down, 1 from the third to the fourth. Flame, let it cool down, and the last one. So 2 from third to the fourth. How many times did we flame here? 10 times, versus here only 4 times. So that's why quadrant is more efficient, versus the radiant is not cost effective because it's very time consuming. We don't usually do that. For the practicum, you might get either one, but just remember that the more efficient one is quadrant, because it's not time consuming and it's cost effective. Now we have utilized, what is the purpose of the plate? Why do we use plate?
Because of the diameter of the specimen.
The surface area, right?
Surface area.
Utilizing surface area for growth of the organisms, and then we might ask, what is agar made up of? Okay, and why don't we use? Is it a solidifying or is it the nutrients? It's solidifying. What is the nutrients? Peptone or beef extracts, amino acids, and then why do we use seaweed extract but not gelatin?
Doesn't melt.
Because it melts at what?
Slowly.
43. Oh, yeah, above 43 degrees.
Okay, so its melting point is 42 degrees centigrade. Why 42 is so important? It's above the incubator temperature, which is the same as the body temperature, 37. So at 37 degrees, it's still solid, versus gelatin is going to be liquid at room temperature. And also, gelatin can be broken by some organisms. It can be digested. So these are the type of questions that you might be asked if you get these. Okay, proper inoculation of the agar dish. If you get this, it's going to be continuous. So the same kind of questions will apply. Proper inoculation of the broth. Nutrients broth, what is the purpose? To grow in broth?
To grow in the agar plate?
No, the broth, the nutrients broth, the liquid.
The turbidity.
Well, that's the
Motility.
results, but why do we use broth but not plate, for example? Or why do we use broth instead of slant?
Motility.
Motility is for the deep. This is to grow large numbers in a small area for DNA extraction and for biochemical testing, PCR and things like that. So broth is usually that's the reason. And then it's made up of peptone extract, or beef extract, and so forth. Okay, purpose of the deep, motility and
aerobe or
okay, so oxygen requirement, so obligatory anaerobes at the bottom, facultative anaerobes. Where do they grow?
In the middle.
Middle, and then microaerophils right under the surface. How about slant? Purpose of the slant?
to save surface area and see the growth.
Okay, so both oxygen requirement and the surface area. Okay, for the heat fix smear, you're going to be smearing, and you're going to say, okay, I have to wait for it to air dry. We will fast forward you and we say, imagine it's air dry, what do you do? And you do heat fix. But we'll ask, which stains do we not heat fix? Capsule or negative stains, right? In negative stains, do we stain the actual organisms or the background? The background. And we do not rinse with water. So heat fixing is to adhere the organisms to the slide. If we don't heat, if we are rinsing with water and we don't heat fix, they'll be washed off. So because in negative stain or you know those acid stain, not acid fast, acid stains, we don't rinse with water. We don't need to adhere, and we do not need to do heat fixing. Okay, so once you go to gram stain, for all those other ones, you can utilize any organisms, and all the organisms that you have tonight, you will have them for that day. So you can utilize any organisms, however, on broth, on plate. But for the staining, you have to utilize the correct organism for that staining. For example, if you get gram staining, you need to utilize either gram positive or gram negative. So give me an example of a gram positive. Staph aureus. Okay, so basically you need to utilize that, and you need to know an infection of Staph aureus, which is any infection of staph.
Cellulitis.
Okay, so it can cause pimples, styes. Basically, we can have folliculitis. Basically carbuncles. You have the staph, scale skin syndrome, or toxic shock syndrome. You have so many things with this, the MRSA, and so forth. And a gram negative, E. coli, diarrhea, right? So if you get gram staining or any of this, you're gonna smear. Say I'm gonna air dry. We say it's air dry. You're gonna heat fix. Then you're gonna add your crystal violet, and you're gonna say, okay, I have to wait for 1 minute. We say, okay, imagine 1 minute is done. You rinse. You add your iodine. So if you need to say it, we fast forward you. Okay, and then you just blood dry, and then because of the staining, it's long, so the questions are going to be only what is the infection, is it gram positive, gram negative, like that. Okay, acid fast. What is the? By the way, for gram staining, what is the virulent factor we're going to be looking at? Okay, what type of cell wall? The peptidoglycan, right? So all of these stains are to identify a virulent factor, a structure that's a virulent factor. So acid fast, what virulent factor? Mycobacterium, and which virulent factor? Mycolic acid. Infection? TB, okay, and basically you're gonna identify that. So the same thing, but the same thing you're gonna just again smear, say we'll fast forward you heat fix, then you're gonna put it on, that water doesn't need to be boiling. You add your paper towel, you add your carbofusion, say 5 minutes, we say, imagine it's done, you rinse, and the same things. So we will fast forward you and then ask a couple of questions. How about endospore, virulent factor? Usually the names are endospore, right? Okay, organism.
That was a trick question.
Organism, bacillus, infection with the bacillus, endospores, anthrax. Okay, and the same kind of concept again. You do the smearing, air drying, heat fixing, and the malachite cream and all the other stuff. Okay, cross out negative and put capsule instead. So for capsule, you're gonna smear again, and you're gonna let it air dry. Say air dry. You say okay, it's done. Then you're gonna add your, after you air dry, do you heat fix?
No.
No. So make sure that you do not heat fix, and then you add your primary stain, your crystal violet. You say 1 minute. You say okay, it's done, and so forth. You're going to be rinsing with copper sulfate, right? What is the virulent factor? Capsule, in the organism, Klebsiella infection, pneumonia. Okay, and so capsule is for adhering and so forth, and then endospore is resistant, and basically dormant, and mycolic acid, slow grower. Okay, if you get hanging drop or wet mount, you're gonna get one of them. Purpose, motility. So again, by the way, for capsule, you have to get Klebsiella pneumoniae. If you get acid fast, you have to get Mycobacterium smagmatis, endospore, bacillus. So take the right organism. And then for hanging drop and wet mount, it was motility, Brownian movement, true motility. Is there any clinical purpose for either one of them? Yes, vaginal, bacterial vaginitis with the whiff test or clue cells and so forth, so you'll do one of them. And then for proper use of microscope, it's the oil immersion. You're gonna have those already prepared slides, the ones you used first week, those brown boxes. It had fungi, algae, bacteria. So oil immersion, which organisms are you gonna use? Bacteria for the oil, not the other ones, only bacteria. Please use the bacteria that you know an infection on. So you're gonna focus, you can add the oil and then focus and then say, okay, it's ready, and then we'll just look and ask you for the morphology. So for example, if you take Staph aureus, you say, what is the morphology? You say cut size, grape clusters, and then we say what is the type of infection, MRSA, folliculitis, any of those, and then that's it. And then the proper demonstration of Petri dish plate pouring is going to be in week 2. You had the soft agar. Remember, you flamed, you poured in the empty, plate. You flamed and then closed. And then the questions would be the same as, what is agar made up of? What is seaweed extract? Why do we utilize that? All the same kind of questions. Okay. So on the back, you have the rubric. So this is going to be out of 20. Each section is 5 points, but we'll adjust it for 65 points. That's the total for your practicum. So 5 points for safety, lab coats, right shoes, make sure hair is tied back, make sure you wear gloves, clean before and after. You don't leave your Bunsen burner unattended, or basically the flame unattended. You don't put your test tube on to fall down and break. You need to have a test tube holder. So all the, basically safety issues. That's going to be 5 points. Flaming the slide. I had somebody yesterday flame the slide with their bare hands. You need to utilize tweezers and things like that. So those are all under safety. So the second and third, so 10 points is 5 points of it is for the steps done correctly, and 5 points is for how confident you are when you're doing it. Okay, do I use malachite green or crystal violet? Do I use methylene blue? Do I use this and that? So if you're not sure, then that's the other part. And then finally 5 points for the questions. Okay. So now, does everyone feel better about the practicum? Again, when you come back on the 9th, Dr. Rafiq is going to be here. I'm not here, but you're going to just practice, because it will show. It's better if you practice rather than just watch the videos.
The 9th is a Tuesday or a Thursday?
The practicum is on Thursday, but Tuesday I won't be here. Because if you remember, I'm here on Tuesdays, Dr. Cindy is here on Thursdays.
Mm-hmm.
So on Tuesday I'm not here. You're going to practice with Dr. Rafiq. On Thursday, Dr. Cindy will be here.
Okay.
For your practicum.
And then we're coming back to read these results on which day? To the 4th?
Okay, so these, any questions on this? On the practicum? The reason I explained it, usually I do this on that Tuesday, week 9, so on the 9th. But because I am not here, I do it tonight, okay? So that you don't have any problems on that night, and you can just practice. Now, this week is going to be on your unknown. You each will get one unknown. In your incubators, you'll find a plate. If you're group 1, you'll take plate 1. If you're group 2, you'll take plate 2, and group 3 will take plate 3. And you need to figure out what your unknown is. It's not going to go beyond the 8 or 9 organisms that you did this term. Which organisms did you work with this term? Which bacteria? E. coli. Anything else? Bacillus, Klebsiella pneumoniae, Staph aureus. How about for acid fast? Mycobacterium smegmatis. Did you work with Pseudomonas aeruginosa? Pseudomonas aeruginosa, Strep pyogenes. You did, Serratia marcescens, really, Faecalis, not very much. Bacillus. So Faecalis and Faecalis is not going to be a part. Serratia is not going to be a part of it. But these other 7, one of these organisms is going to be yours. Okay. So basically, what you're going to do, and let me bring it up. So this lab is going to actually cover your that missed lab that you that got thrown away. So let me just. Okay, so on your lab manuals, remember week 2 there were 3 labs: the blood, the mannitol salt, and the EMB, and those got thrown away. So you're going to make that up on that 4th of January to make up for your 4th of July. Okay, so right, everyone remembers? Okay, so these 3 labs you did not submit to Dr. Cindy. So you're going to do them tonight. However, these 3 labs are part of your unknown, to identify your unknown. So because these are special medias or selected medias that will show you differential media, your unknown, how it looks like. So for example, I'm going to start with the blood agar. So let me put on my gloves. So on the blood agar page, I'm going to start with the bottom and we'll move up. So the first one is your blood agar. It's on page 99 of your lab manuals. And you're going to take one of these blood agar. Now let me show you the difference between blood and mannitol salt. So, EMB. So this is the blood. This is EMB. You see the difference? So don't mix them because they're not labeled. So you label yours, okay? And once the blood stays a little bit in the incubator, it will be very close to EMB as far as the color. So you're going to take your blood on page 99 of your lab manual, or actually page 100 of your lab manual. You have under the organisms, what you're going to do is you're going to take your plate, your blood agar plate, and you're going to divide it into 4 sections. One section you're going to inoculate with Strep pyogenes. Okay? One section you're going to inoculate, and you're going to just do either a tornado or just one streak. Okay? One section Strep pyogenes, one section Strep sanguis, one section Faecalis, and the 4th one is going to be your unknown. That's on the plate with your group number. Instead of, it says patient A, you're going to do your unknown. Okay? And then you're going to parafilm, invert, and incubate. When you come back on the 4th of January, you're going to actually read these results and put them on page 103. Okay? But on Thursday, you're going to put your results here, just for your unknown. So that corner, that's the unknown, you're going to put it here. The other ones, and also your unknown, you're going to put in this on the 4th of January. So we're just, instead of doing 2 plates, you're just doing 1 plate. Okay? Remember when you did the alcohol, when you put your thumb, some of you must have seen that flesh-eating bacteria, right? The beta hemolysis. So strep pyogenes causes necrotizing fasciitis. It causes that clearing, complete clearing of the blood, beta hemolysis. Fecalis does nothing, gamma, and then sanguis does alpha, which is partial. So you're going to see what your unknown. One of your unknown might be strep pyogenes, so it might do that. Okay. The next one is going to be mannitol salt on page 105, and your mannitol salt is these plates, the pink ones. So again, for your mannitol salt, you're going to divide it into 4 sections. And on page 106, one section you're going to streak all of these aseptically, please. One section you're going to streak with Staph aureus, one epidermidis, Staph epidermidis, one is going to be E. coli, and the last one is going to be your unknown. Okay? And then again, gram-positive, this is selected for gram-positive, epidermidis, aureus, all of them will grow, E. coli will not, it's high salt concentration, gram-negative will not grow. But Staph aureus will produce those yellow colonies, does the fermentation. So again, the same thing, on 4th of January, you're gonna put your results on page 109, but on Thursday, you're gonna put your results on here for unknown only. Okay? And then finally, the last one is your EMB on page 111. And you're going to divide your EMB again into 4 sections. One is going to be E. coli, one Enterobacter origin, one Pseudomonas aeruginosa, and the last one your unknown. Instead of sample P, you're going to do unknown. Okay? And you're going to parafilm, invert, and incubate. On Thursday, you'll read results. Do not throw away. We're going to keep them until January 4th to do these 3 labs and submit. Okay, so that's the first part of your unknown. Once you inoculated all of your 3 plates, we're going to start doing your staining on your unknown. How many different stains did we do this term? 4. Gram staining, acid fast, capsule, and endospore. So you're going to do all 4 stains on your unknown and then put your results here. So hopefully by Thursday when you come back, once you look at your plates plus your staining, you should know what your unknown is. If not, there's catalase and oxidase test that Dr. Cindy will show you to figure out what your unknown is. Okay? I know I talked a lot tonight, but I had to go over like 3 different, this week, plus your practicum, plus that January 4th. So I had to do all of those together. Okay, any questions? Okay, thank you. Go ahead and get started. If you want these plates,
Yes,
oh, be
please.
careful.
Do we turn this on?
No, you're gonna upload. So that,
Those 3.
Thank you.
yes.
But for this one, the only thing they're gonna like put on this one is the unknown. All the other ones are gonna go on like the lab.
The actual lab
Yeah.
itself.
Wait, so today we're doing unknown?
We're doing everything. Because she wants us to divide it in 4. So we're doing it. That's the thing. Like, why can't we just do it on this first day? Because we're doing.
Wait,
We're doing. We're doing
I thought
that
this-
and this. 'Cause we're doing that, but instead of putting the
Okay.
unknown A, we're gonna do. I mean the patient A. We're gonna
do
do
we all
our.
have the same unknown or do we have a different unknown?
Our group has the same unknown.
So we can split up these plates because
Yes.
we don't. Okay. Okay, I thought we were like all had to do it. Okay, are you kidding?
All the organisms you need also are in the test tube except your unknown on a plate.
Do we have any data that we can see today? That we did?
No.
Is our, what do you call it?
Pardon?
Our responses? They're due tomorrow?
Yes, but the peer responses aren't due until not this Sunday, like January.
But our
Sunday.
actual responses.
But the actual responses are due tomorrow.
Is due tomorrow.
I know.
Like, hold on.